It's the scenario every neurologist and ophthalmologist dreads. A patient rushes in terrified because one eye has just suddenly gone dark.
A stroke of the eye, central retinal artery occlusion.
And for years, we've looked at our success in treating brain strokes with clot-busting drugs and thought, why can't we do that for the retina?
Well, today we have what looks like a definitive answer.
And unfortunately, it is not the one we were hoping for.
It really isn't. We're looking at a trial that many of us thought, or at least hoped, might change the standard of care. Instead, it seems to have, well, slammed a door shut.
Welcome to the Weekly Neurology Deep Dive. This program is produced by Amer Gavanini. I'm your host, and I'm here with our resident expert to break down the latest practice-changing literature.
It's a pleasure to be here.
Now, before we get into the data, we have to state our standard disclaimer. This deep dive is for educational purposes only.
There's no commercial goal here. We're summarizing complex data, and we really do recommend you read the full paper yourself.
Absolutely. So, the paper we're discussing is titled A Randomized Trial of Tenecteplase in Acute Central Retinal Artery Occlusion.
It's by Ryan, Jerstead, Scranland, and their colleagues.
Published in the New England Journal of Medicine on January 29, 2026. This is the TenCRAOS trial.
A major contribution. We've been waiting for high-quality data on this specific question for a long, long time.
I know our listeners are busy clinicians, so let's start with the executive practical summary. What is the bottom line here?
The question was pretty straightforward. Does intravenous tenecteplase at a dose of 0.25 milligrams per kilogram improve visual outcomes if it's given within 4.5 hours?
And the answer?
A clear no. The trial found no significant difference in vision recovery when you compare it to just aspirin alone.
And there's more to it than just a lack of benefit.
There is, and this is crucial for neurologists. The drug was associated with severe safety concerns, including one fatal intracranial hemorrhage.
So the final recommendation?
The findings do not support the routine use of systemic thrombolysis for this indication.
That is a very clear, if disappointing, takeaway. Let's unpack this because the why and how are fascinating. First, the background. Central retinal artery occlusion. An absolute emergency.
It is. It causes sudden, painless vision loss in one eye, and if you don't get reperfusion quickly, it leads to permanent blindness. The retina has an incredibly high metabolic demand.
And the central retinal artery is an end artery, almost no collateral flow.
Right, so the analogy to an ischemic stroke has always been there. We think there's a clot, it's causing ischemia, so why not use thrombolysis?
We've tried before, haven't we, with other drugs?
We have. Previous trials like THEIA, which used intravenous alteplase, and EGL with intraarterial lysis. They all showed mixed or negative results.
But the world of stroke neurology has shifted. We've largely moved on from alteplase.
We have to Tenecteplase. It's a better drug in many ways. Greater fibrin specificity, a longer half-life.
And it's just so much easier to give. A single intravenous bolus instead of an hour-long infusion.
Exactly. So the logic for this trial, for TenCRAOS, was compelling. Maybe the other trials failed because the drug wasn't quite right or it was too slow. Maybe this better, faster drug was the key.
That was the hope. So let's get into the design of the trial.
It was a phase 3, double-blind, double-dummy, randomized controlled trial. Very rigorous.
Conducted where?
At 16 sites across 6 European countries.
I always appreciate a double dummy design. It shows real commitment to eliminating bias.
It does. It meant every patient got 2 treatments to maintain that blind. One group got active intravenous tenecteplase plus an oral placebo.
And the control group.
They got an intravenous placebo plus active oral aspirin.
So who got into the study? What were the inclusion criteria?
They included adults with acute, non-arteritic central retinal artery occlusion. And the time window here is the critical piece.
How fast did they have to be?
A strict 4.5 hours from symptom onset.
Which, mirrors the standard intravenous thrombolysis window for acute ischemic stroke.
It does. They also had a severity criterion. Patients had to have a best-corrected visual acuity of 1.0 log-AMER or greater.
And for those of us who don't think in log-AMER every day,
that's equivalent to 2200 vision, or worse. So these were patients with profound vision loss.
And I assume standard stroke exclusions applied. Neuroimaging for everyone?
Absolutely. Everyone had imaging to rule out any other intracranial events before they were randomized.
Okay, so we have the 2 groups. Group A gets tenecteplase, Group B gets aspirin. What was the primary endpoint they were looking for?
Vision recovery at 30 days.
And how did they define recovery?
As achieving a best-corrected visual acuity of 3.0.7 log-AMER or better. In standard terms, that's about 2100 vision or better.
That seems like a reasonable bar. moving from 2,200 or worse to 2,100. That's a clinically meaningful improvement for a patient.
It is. It represents a change of at least 15 letters on the vision chart. It's definitely meaningful.
All right, let's get to the results. How many patients did they end up with?
They included 78 patients in the final analysis. The baseline vision in the group was, as expected, very severe. Many could only count fingers or perceive hand motion.
And here's the part that really stood out to me: the time to treatment.
They were incredibly fast. About 69% of patients received treatment within just 3 hours of symptom onset.
That is a remarkably fast response time, so we can't blame the system here. We can't just say, oh, they got the drug too late.
No, not at all. Most of them were in that super early window.
So, did it work? Let's hear the primary endpoint data.
In the Tenecteplase group, 8 out of 40 patients achieved that recovery endpoint. That's exactly 20%.
And in the aspirin group?
In the aspirin group, 9 out of 38 patients recovered. That comes out to about 24%.
Wait, the aspirin group actually did numerically better.
Numerically, yes, about 24% versus 20%.
Statistically?
Statistically, the difference was not significant. It's essentially a wash. There's no benefit shown for Tenecteplase.
That is just sobering. The better drug, the fast delivery, the rigorous design, and it couldn't beat aspirin.
It's a tough result to see.
What about any of the secondary endpoints? Was there a glimmer of hope anywhere else?
They looked at other thresholds, like achieving 0.5 log MAR, which is about 20/63 vision. Again, no substantial difference. About 20% versus 18%.
And mean improvement. Visual fields.
Nothing. No significant advantage for thrombolysis anywhere.
What about that big group treated within 3 hours? Surely if you treat that early, you have to see some kind of signal.
That's what you'd think. The investigators did a subgroup analysis on just those patients. Even in that super early window, no benefit was observed.
Which brings us to the other side of the coin. If a drug doesn't work, that's one thing. If it doesn't work and it's dangerous, that's a whole other conversation.
And we have to have that conversation. Let's talk about safety.
This is what every neurologist listening needs to hear.
The adverse events were, as you might expect, higher in the Tenecteplase group. But there was one event in particular that changes everything.
What was it?
There was one fatal intracranial hemorrhage in the Tenecteplase group.
A fatal brain bleed. That is the nightmare scenario. We're trying to save vision in the eye, and the patient dies from a brain hemorrhage.
It really highlights the risk we take with any systemic lytic. The dose, 0.25 milligrams per kilogram, is the standard stroke dose. But the risk-benefit calculation is completely different here.
How so?
In a brain stroke, the brain is the target.
Oh.
We accept the risk of hemorrhage to save brain tissue. Here, the eye is the target, but the brain is still exposed to all the risk of the drug.
So why did the bleed happen? Was it just bad luck?
Not entirely. It seems to be related to the underlying pathophysiology. We know from other studies that about 20 to 30% of these patients have concurrent silent brain ischemia.
So there are little strokes happening in the brain that aren't causing symptoms, but they're on an MRI.
Precisely.
Oh.
You give the Tenecteplase to clear the retinal artery, but it circulates everywhere. It finds that fresh, silent ischemic damage in the brain.
And it causes a reperfusion hemorrhage.
Exactly. The patient in this trial had ongoing cerebral ischemia that wasn't apparent clinically or on the initial CT scan. The drug caused multiple intracerebral hemorrhages.
But that is a devastating outcome. And frankly, it changes the entire calculus. If the benefit is zero and the risk of death, however small, is real, you just can't justify it.
That's the conclusion.
Let's zoom out a bit. How does this finding fit with what we already knew from the literature?
It aligns perfectly with the HEIA trial with alteplase and the EGLE trial with intraarterial lysis. We now have high-quality evidence from multiple trials, and the conclusion is consistent.
Systemic thrombolysis within 4.5 hours just does not work for this.
It is not.
Which brings up the big question, why? Why does it work for a clot in the middle cerebral artery, but not one in the central retinal artery?
There are two main theories discussed in the paper. The first is pure anatomy.
The end artery problem.
Yes. The brain has the circle of Willis. It has collateral flow that can keep tissue alive for hours. The retina has almost nothing.
So while our time as brain window is 4.5 hours, or even longer, the time as retina window might be. What?
Some animal models suggest irreversible damage happens in as little as 100 minutes. If the tissue is dead by minute 105, giving a lytic at 3 hours is just too late.
In the second theory,
The composition of the clot itself. Emboli to the eye often come from carotid plaques. They might be calcific or cholesterol crystals.
Hollenhorst plaques.
Right. And thrombolytics, like Tenecteplase, work on fibrin. If the plug is made of calcium or cholesterol, the drug just flows right past it.
So you're exposing the patient to all the bleeding risk, but the target clot itself is resistant to the drug.
That's the theory, and it makes a lot of sense.
So let's summarize the clinical takeaway. A patient comes into your emergency room next week, central retinal artery occlusion, 3 hours from onset. What do we do?
You do not offer intravenous Tenecteplase. The results of this trial are clear. The risk of a fatal brain bleed outweighs a non-existent benefit.
It's a hard conversation to have with a patient who is losing their vision.
It is, but it's the evidence-based one. We manage them conservatively, we look for the cause to prevent a future stroke, but we don't reach for the thrombolytic.
Let's do a final recap for everyone listening before we sign off.
The trial was TenCRAOS. It compared intravenous Tenecteplase to aspirin for central retinal artery occlusion.
The result.
Tenecteplase within 4.5 hours did not improve visual outcomes.
And the safety.
It was associated with increased risk, including a fatal intracranial hemorrhage.
So the practice point is,
Do not use thrombolysis routinely for this indication based on this data.
I want to leave our listeners with one last provocative thought. If intravenous lysis fails within 4.5 hours, and even intra-arterial lysis fails, is the time is retina window just too short for any hospital-based intervention?
That is the big question, isn't it? By the time a patient realizes what's happening, gets to the hospital, gets triaged, that 100-minute window might already be closed.
It makes you wonder if we're treating the wrong mechanism, or if the only possible solution is something that can happen before they even reach the hospital. But that's a discussion for another day.
Indeed. For now, we follow the evidence we have.
Thank you for that excellent analysis. And our thanks to our producer, Amerga Manani. We will see you in the next deep dive.